I'm going to kick off by talking about, I guess, one of the older forms of parcellation, about parcellating the brain according to histological and microstructural architecture. And the reason I'm going to talk about this is because history is important and informs us about where we're going. So one more question for you all then. This question is, why parcellate the brain? And I presume that everyone in this room has thought about that because you're all here. To find functional units, for network analysis, for fun, yes! Right, for comparison, the size of that laugh suggests that not everyone thinks it's fun yet. Right, so for the purposes of comparison, I'm not sure what multiple comparison problem means in this context. Interesting. Right, yes, topography, dimension reduction, to turn an ill-posed problem into a well-posed problem. Big claim. We have cross-species homology. That's one that's very important to me. To address the limits of brain registration. There's like a resolution issue to be thought of here. Okay, great. So same thing. Keep those questions in mind. Let's see what's going on. So here are some of the things that I thought about when I was thinking about why parcellate the brain. So you've already mentioned some of these. So to localize function. This can be to localize function through the parcellation or just have a parcellation in order to put your functional MRI data onto. To create some kind of common language to understand the brain. Obviously, we have some common languages already. This is just another one. Another way to understand individual variation, which I think several people said in different ways. To compare individuals or groups. To compare species. I think we covered all of these already, basically. Okay, so here are some types of anatomical parcellation then. We have macrostructure, so sulcal/gyral anatomy. This becomes less and less fun as you go more and more non-human and further away in evolutionary terms from the human brain. So, for example, some of the delineations that exist in the human brain don't exist in the monkey brain even. Microstructure or cytoarchitecture, the architecture of cells present in the cortex. I'm going to talk about this as a starting point. Myeloarchitectonics, the structure of myelin. Structural connectivity. And then, of course, functional connectivity. So I'm going to talk least about functional connectivity because lots of other people are going to do that in this session. But what I am going to talk about is why and how to use microstructure to parcellate the brain. And I'm going to try and do this extremely rapidly to leave as much time for debate and questions as physically possible. Okay, so one of the reasons that I said already is that macro anatomical landmarks are problematic. They don't align well with really anything. Even in the human brain, they're extremely variable. For example, I have a paracingulate sulcus in my left hemisphere, but not in my right hemisphere. What does that mean? It depends on who I am and what those areas of my brain are doing and how they line up. Microstructural maps, on the other hand, are arguably much more precise. They're arguably objective, and they're arguably reproducible. You can decide for yourselves whether you agree with that. You have to agree with this because it's true. It relates to function. That is the fact that there is an organization of neurons in the brain has something to do with what they're doing. That isn't to say that it's the be-all and end-all, but it relates strongly to the function of different brain areas. And I'm going to give you a few examples of that. So here's one example. This is an old piece of work now. So in this study, which, as you can see, this is like a drawing, old school style of the brain, what they did here is they drew a map of myelo-architecture. That is how the myelination density of the different layers of the cortex varied. And they lined it up with brain stimulation studies. So putting an electrode into patients' brains as and when the opportunity arose to stimulate. And what they were looking for was whether there was a good alignment between the architecture at the cellular level and the functional properties. So the first one was the map of myelo-architecture that you can see here, and then what they did is they went through and painstakingly stimulated in these neurosurgical patients, and they made notes as to what happened when they stimulated in these areas, and then they looked for correspondence between them. And at least in some areas, they found really good correspondence between this myelo-architecture structure and the functional boundaries, so the pink areas there. To some extent, you can see that there are clusters of these different stimulation patterns that are clustering together that line up relatively well with the myelo-architectonic boundaries. So, for example, you can see that there is a very, very sharp boundary between these circles and these circles here. These squares and these circles here for the hind limb and the forelimb areas of stimulation. Oh, this obviously wasn't in patients. My mistake. I don't know any patients that have hind limbs and forelimbs, so this was an animal study. The previous study was a patient study. Okay, so that was myelo-architecture. That's the myelinated structure of the cortex. This is cytoarchitecture. You probably already know this if you've done a neuroscience 101 course of any sort. The cortex has, in general, 6 layers. And those layers have different cell types, which you can see on the left-hand side. And those pertain to the ratio of inputs to outputs in those different cortical layers. So, for example, layers 2, 3 is mostly cortical inputs. So these layers, you can see them by eye. If you take a slide sectioned through the brain at a particular level and you stain it with something that shows up cellular architecture, like a Nissl stain, then you can see this kind of architecture. And yet, it corresponds really well to how the inputs and outputs of that area of cortex are arranged. So the fact that that cellular architecture exists gives us some clues as to function. And the most famous of all the people who looked at this in order to parcellate the brain was Brodmann. So what he did was went through section by section and he looked for boundaries where there was a change in the cytoarchitecture. So the cytoarchitecture isn't the cell types in each area. There can be the same cell types in multiple areas. It's their arrangement. And what he did was looked for boundaries where that arrangement changed, and then he marked on those boundaries, and he drew these hard boundaries. Now, most of Brodmann's work was done in a single brain per species, and that's worth mentioning. So somebody mentioned inter-individual variation before. This doesn't necessarily take that into account. But here are some other examples of how that relates to function. So, for example, in agranular cortex, such as primary motor cortex, what you see is that there are these giant cells in layer 5 of motor cortex. Now, these giant cells are called Bett cells, and they have these huge myelinated axons. And it turns out that that does relate to function. So the larger your axon and the more well myelinated it is, the faster it can fire. And if you're motor cortex, the one thing that you need to do is get signals down into the spinal cord and out to the limbs quickly. So here's one example of structure relating to function. Here's another one, primary visual cortex, often called striate cortex because it has this stripy feature, which is particularly well visualized when stained with cytochrome oxidase. has this extremely complex layer 4, which has these like sublayers 4A, 4B, and 4C. Why? Because it has to receive all of these complex inputs. And here's another example, which is prefrontal cortex, which is known as granular cortex, which has these granular cells in layer 4, again, to allow it to receive inputs and be an area of higher order association cortex. So there's all kinds of examples where just cell types recapitulate function in the brain. So as I said, Brodmann came up with these hard boundaries because he had one case at a time. But in an ideal world, we would see how that relates from person to person. So in the modern world, what happens is that we can plot these I say we, none of this work in this talk is mine, so I can't take any credit. So what we can do is we can plot here a metric of the density of different cell types in the cortex, and we can look for, again, these boundaries, these sharp transitions, using a mathematical model now instead of using our eye, as Brodmann did, and then we can look at that across multiple subjects. So when we do that, what you can see is that the boundaries become much less sharp. So here, for example, is area 10, the frontopolar cortex, and here is here's a rendition of the overlap from 10 individual subjects. So the red is the most overlap, where area 10 exists in all 10 subjects, and the cooler colors are the areas of least overlap. And what you can see now is that there's a fuzzy boundary, even when transformed into a common space. So if we use these techniques across subjects, then we can start to see that the boundaries maybe are not quite such hard and fast boundaries. And so some of the questions we'll be attempting to ask in this session are how do we deal with that? You know, how if I now want to map this onto my brain, or I want to localize a function of my brain, how am I going to go about doing that? Let's revisit myeloarchitecture for a minute, just so you can see what I mean. So I spoke about that earlier, right at the start. So the stain that I've been showing you the most is the Nissl stain, which stains for cell bodies. So this is the standard cytoarchitecture stain. The stain in the middle is the myelin stain. And the myelin stain is as you can see, stains darkly for myelinated axons. And you can see those axons in play on the right-hand side, which is our very, very famous stain, the Golgi stain, which is the one that Cajal used to visualize all of his neurons. The Golgi stain captures every aspect of the neuron, so the axon and the dendrites and all the branches of it, as well as the cell body. So there's a number of different stains that can be used, and it's good to know that. And what you can see is that myeloarchitecture also has gradients and differs from area to area. So I'm just showing you a number of cortical areas. Helpfully, this paper does not use the same numeration as the cytoarchitecture papers. So these numbers are not very helpful. But if you were to go through and line them up one by one, you would see some correspondence between the two. What's interesting about this, since everyone else in this session is going to talk about MRI, is that if you look at how the definition of something like cortical depth fares when using something like a T1-based MRI imaging, and how it stacks up against the same definition being made with a myelin stain or with a cell body stain, I hope that what you can see from this study at least is that the thing that correlates the best is the thing we talk about least often, which is the myelin stain. So there's actually pretty good correspondence between the myelin stain and the T1 imaging here. And so it's worth bearing in mind, I think, that although cytoarchitecture is considered a kind of gold standard here, that there are actually a lot of ways that we can look at structure, and there are a lot of ways that we can parcellate the brain, and we might want to take those things into account. Of course, not everyone can do this. For example, if you have participants coming into your lab, I do not recommend that you myelin stain them. Thank goodness someone laughed. All right, so of course I'm almost out of time, and I'm about halfway through my talk, so let's see. So we also have connectivity. This was done by dissection, and then later in animal studies it was done with tract tracing. So tract tracing is where you put a dye into the brain, and you have it be the kind of dye that can either be carried forward or backward in terms of connections. This relies on the intrinsic properties of the neurons, and it requires the neurons to be alive. And then it requires you to do some staining or some visualization under a microscope afterwards. It can only be done in animals. As a result, almost everything that we know about the connectivity, the structural connectivity of the human brain, is extrapolated from monkeys. We don't have any of this information directly in the human brain. So when we say that a connection exists between two areas of the human brain, unless you can see it at this gross level with blunt dissection by freezing and then peeling apart the brain, which just shows these major tracts, if you hear somebody say that a connection exists between two areas, we don't know that for sure. However, diffusion imaging gives us a very good clue. So again, I'm giving you this talk in part to get you thinking about how those MRI measures might relate to things that we think or know are real. So here's an example of that under the microscope in a monkey. Yes, it's directionally specific. So anterograde tracers show you the connections from one region to another. Retrograde tracers show you the connections that are coming into a region. MRI can't give us that directional specificity. So there are caveats associated with our MRI, but on the other hand, we don't have access to this in the human brain at all. This is a monkey brain that I'm showing you. These tract tracing connections have been used to come up with connectivity fingerprints, which can allow us to start to parcellate regions of the brain based on their connections to a number of other regions and the pattern of those connections. So what you can see is that for these two areas, in the prefrontal cortex on the left and in area 9 in the premotor cortex on the right, there are different patterns of connections to other cortical areas, allowing us to come up with a parcellation based on the tract tracing connections of these areas. And what you can see here is that if these are submitted to a dimensionality analysis, then you can see that they end up with clusters of brain regions. And these clusters of brain regions, I had circles around them, I don't know where my circles have gone, but they're in different subregions of the prefrontal cortex. For example, all of the orbitofrontal regions cluster together. And so then we can come up with these connectivity parcellations. Post-mortem tract tracing, on the other hand, which could be done in the human brain, is just on the order of a few millimeters. So we can never look at those long-range connections directly in the human brain. Okay, so in the last couple of minutes then, I want you to be aware of some studies that have gone on. These slides will also be posted online, I think, if you want to scrutinize them in any detail. So people have started to look at whether you can predict one of these things from another one. This is really beautiful work by Helen Barbas showing that cytoarchitecture predicts connectivity. So what she did here was looked at cytoarchitectonics, and then she looked at the direction of connectivity and was asking the question of whether one was related directly to the other. And what she showed was that so what she did is she mapped all these cortical areas in terms of the complexity, so how laminate they are. So what you can see here from left to right is that there's a from agranular up to disgranular and then to the eulaminate areas, you see more and more laminateness, amount of complexity in the laminar structure. And that relates very well with areas of a more complex laminar structure showing these feed-forward projections to areas of a less complex laminar structure. And that the model that they came up really well predicted the connectivity of these areas. So there is a relationship at the cytoarchitectural and tract tracing level between those two types of parcellation, potentially. In addition, neuron size, plotted on the bottom here, across cortical layers, predicts a macro scale connectivity. So now looking at an MRI measure, so diffusion tractography connectivity, particularly the size of the neurons in layer 3. So this is histological data plotted onto a brain and then correlated against the tractography findings. And what you get here is a positive correlation between the size of the neurons in layer 3 and the connectivity, layer 3 being a major area of connectivity. It also correlates, there are also strong correlations between dendritic spine density and structural connectivity. So in this case, they didn't have these data for every brain region, so there's a couple of caveats with this study as well. But what they did was they lined up the layer 3 spine data onto these areas and then correlated some of the features like spine density or dendritic tree length with the macro scale degree. So that's a graph theoretical measure of the amount of connectivity between two areas, which we'll go into in more depth in one of the other talks. And there's a positive correlation between those things. So we can start to relate some of these things to each other. I quickly want to say a lot of these dendritic spine studies in humans are done with the Golgi stain. The Golgi stain is amazing, it's beautiful. It doesn't pick up every part of the dendritic tree. If you want to do that, you have to take an animal and you have to fill the neuron post-mortem with lucifer yellow very slowly and carefully over time. This is my work. It's the only thing I'm going to show. Or you have to have an animal that has fluorescent staining, so it has like GFP or YFP in its neurons in order to really be sure about this. So we still need animal studies to verify some of this work. And this dendritic spine density is dynamic information. It changes, and it can change with things like the gray matter volume as measured by MRI. I think that's important. Cytoarchitecture predicts connectivity then not only at the microstructural level, but also at the MRI level, giving us a metric that maybe can span resolutions. And I think that this is really an important feature as well. So I'm going to leave you with a couple of limitations. Microstructure is incredibly time-consuming. I do not recommend it if you want to get on with your life in a hurry. It takes a really long time to do these microstructural studies, many, many years per brain area in some cases, depending how many subjects you have. It can only reliably be performed post-mortem, so that relies on donated brains or animal brains. And we're still figuring these things out. So those findings that I showed you are very, very specific. They're just about the layer 3 density versus the macroscale connectivity of the whole brain. So it's going to take a while until we can actually line these things up well. And we don't know what that means in terms of comparing across groups and so on. Ooh. Okay, so we have a couple of minutes for questions before we move on to the next talk. So if we have any questions now, you can either just ask them or you can throw them up on this screen if it ever loads. This is where the awkward silence commences.